डीएनए आइसोलेशन के बाद जो अगला मरहला है वो डीएनए की कंसंट्रेशन डिटर्मिनेशन है. मीन्स हाउ मच डीएनए इज प्रेजेंट इन एनी नोन वॉल्यूम, microgram in microliters. For this purpose, we need some reagents, supplies, and we require equipment for a proper usage. The supplies we need, of course, the sample DNA from your any experiment which you want to use further, DNA standards of known concentration from lowest to highest concentration, ethidium bromide stock, ethidium bromide waste container. For self-protection, we need goggles, face shield, and UV blocking purpose, plastic wrap, Polaroid film if you are using a conventional or old-fashioned camera for instant photography, quartz cuvette if you want to use the spectrophotometer, UV spectrophotometer for concentration determination, sterile water, Tris EDTA buffer to make the dilution of the DNA. Equipment required is a camera, digital camera or a Polaroid camera, whatever is available, the spectrophotometer which can read the UV light, and ultraviolet trans illuminator. Besides the DNA samples or DNA standard, we need the ethidium bromide to stain or to dye the DNA. Ethidium bromide, which will be used in the quantitation, is required at 1% concentration. Ethidium bromide is sparingly soluble in water, therefore we dissolve it in 95% alcohol, that is ethanol. 1 milliliter of ethidium bromide is added in 95% ethanol. Magnetic stirrer is used to mix the ethidium bromide in ethanol, and then volume is made up to 100 milliliter using distilled water. Ethidium bromide itself is light sensitive solution, therefore you should store this solution in brown bottles or in foil wrapped bottles. This chemical is highly toxic and is a potential mutagen. Therefore, gloves, goggles and lab coat must be used while working with ethidium bromide. As the ethidium bromide is a mutagenic agent, it should be disposed of properly after it has been used. There are two alternatives to dispose of the ethidium bromide safely. The first one is decontaminating the liquid solutions through filtration or extraction. We can use the filter which can filter the ethidium bromide specifically so that the waste liquid should be clean from ethidium bromide. The second method is the chemical treatment, which needs 24 hours to decontaminate the waste. For this purpose, the ethidium bromide waste is supplemented with phosphoric acid. 25 milliliter of freshly prepared 5% phosphoric acid is added along with 12 milliliter of sodium nitrate. The pH of the solution should be less than 3. This mixture should be kept for 20 hours, and after 20 hours, neutralizing this waste solution of ethidium bromide with the help of sodium carbonate, with the help of sodium bicarbonate, it can be discarded later on.